Good morning, everybody. All right, usual deal. Wait for people to show up. Anybody got any questions in the meantime? Feel free to fire away. Oh, that's super cool, Rose. It's those animations are really, really excellent, and we'll be looking at them for DNA replication and transcription and translation. And even though they're a little bit, I mean, they're not very easy to watch in a way because they don't have labels. They're not very cartoonish. They're very much based on what things actually look like as best as we know it. They are very accurate, and they really do show what goes on again as best as we understand it so far, at least. And so there's none of the kind of risk of fluff and waffle that you find in a lot of YouTube videos where they kind of fudge the facts a little bit. Right, so you have to be often. I'm a little bit leery of using stuff off the internet, but those ones are exceptionally good. The only thing that's a little bit weird is that they have like this molecular noise going on, which I always find a little bit odd. Khaled, about the book. Again, I kind of talked about this in the first class, maybe. Whether or not you use the textbook is up to you. It's very much an individual decision. I'm not going to kind of mandate it. I just provide you the information of what book we would be using and what chapters or part of the chapters would be relevant to this particular course at any given time. So those readings are, or at least the guide to the readings, are in the syllabus section. Yeah, I think it's in the syllabus section. And as for the book, you can get either a 6th or 7th edition book. Obviously, 7th is newer, has a little bit newer information, but nothing that significantly changes what you get out of it. And those you can either get from the bookstore for the new one, which is probably some crazy amount of money, or you can get a 6th edition off of Amazon, eBay, wherever else you'd find used textbooks. You can even rent them as well. I know that some people in these classes have rented textbooks for a pretty reasonable amount of money. I don't know what it would be, like 30 or 40 bucks for a semester, which compared to buying a new one for a couple of $100 is a pretty good deal. So it really depends on whether or not you think you'll get much out of it. And if you do, then it'll be worth getting. If you don't, then don't. The big plus of the textbooks are the questions at the end of the chapters as well, which will help, which if you work through all of those is a very good way to study as well. So that's kind of up to you. Morning, Tanya. All right.
Good morning.
Good morning. Does anybody have questions about what we've covered last class? So would that be really the monohybrid cross and test cross is what we covered last class, I believe. Give you all the opportunity to have a think about that and ask questions if you want.
For for the dihybrid cross, it'll be the same, like for the test cross, the F1 generation is going to mark the dominant.
Ooh, let's not confuse test crosses and mono and dihybrid crosses. They're very different. So a dihybrid cross is exactly the same as a monohybrid cross. Same, and actually it's kind of preempting essentially what I'll be telling you in a little moment anyway. You have the, now let's get this up. Rather than wave my hands around, I can just show you instead. Which one are we at? Come on. No, that's what I wanted. Oh, yeah, she is there. Right, so, pardon me. You will get the same 3 generations in a dihybrid cross that you do in a monohybrid cross. P generation, F1 generation, F2 generation. The differences between the two, well, the similarities, obviously, which is worth pointing out. The P generation have true breeding or homozygous parents. Typically in a dihybrid, we have them both, one parent being homozygous dominant for both genes, the other one being homozygous recessive. Doesn't actually make any difference whether they are or if they're not. You get the same output. Another similarity is that the F1s are heterozygous for all genes involved. And as such, they display the phenotype of the dominant parent or the dominant phenotypes, right? You know, because they're heterozygous. And those are the similarities, right? So the same 3 generations, again, it's a very kind of rigid structure. That's why if you change any of this, it's no longer a monohybrid or dihybrid cross. It's something else. Back cross, test cross, linkage cross, whatever you want to call it. It's not a mono or dihybrid cross because that tells you exactly what's going on. Homozygous parents, heterozygous F1, and then those F1s are crossed to each other and you get your F2 generation, which has the stereotypical ratio, right? The differences between them are we have 2 genes, right? Each of those genes has 2 alleles and 2 phenotypes. So now we kind of essentially we have, rather than 2 different phenotypes present or combinations of phenotypes, we now have 4. Right, so things get more complicated essentially. And also the other difference is that the F2 generation, the ratio is, as a consequence of that, a different one. So now we have 4 different phenotype combinations rather than 2. So monohybrid is 3-to-1. Dihybrid is now going to be something, 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 right? Because now we have 4 different categories. And the ratio is 9-to-3 to 3-to-1. And we'll get to why that is in a moment. So as long as you understand what a monohybrid cross is, really a dihybrid cross is the same again, but now you're just doing it for 2 lots of things rather than just 1. Yeah, that's why it's really important to get the monohybrid first, because otherwise dihybrids is quite difficult to understand. Still, it's relatively simple, but there's just a little bit more going on that can throw you for a loop. Let's see, where me chat session. No, didn't want to do that. Did want to do this. Awesome. Does anybody else have any questions about stuff that we learned last class? Just give you a chance to clear up any confusion or the like. Nope. Okay. So what I want to do today is really finish this up, which shouldn't take very long. But we have time to spend on it if you need that. And then we're going to dive into the essentially probability and statistics in genetics PowerPoint, right? So whether we get that finished all today or not, I don't know. We'll just have to see how it goes. Let's just double check the schedule again. We're actually a little ahead of the curve according to the schedule, because we have this pegged for Tuesday. So we can take our time today, and we can probably on Tuesday, we'll spend the bulk of the time going through chi-square problems, right? Because that's something that is, it's not tricky to get, but there's a bunch of different things going on in there that all have to come together in the right way, right? So it's easily achievable for all of you, but I just want to be very kind of deliberate about how I go through that with you. And we're also going to have a guest lecturer. Tanya is going to show you how to do this in R, because I don't know how to do it in R. So she's going, right, Tanya? We talked about this, yeah? Practice being a professor for a little while. It'd be fun, right? No, maybe not. Perhaps I jumped the gun a little bit there. Because I can't do it now. I'll have to like copy and paste that into a Word document or something, just in case I need it in the future. R is a statistical program, which I showed some of you in the lab. This is what it looks like. It's clunky as hell because you have to type stuff in by hand, which I'm not very good at doing. But it's also super, super powerful for doing statistics. This is doing some proportion testing, essentially. All right, where are we at? So anyway, going by the schedule, we've got a little bit of leeway. Don't forget as well, we've got an invited lecture on the 9th of February. I have the form to upload. I'll do that most likely today, send you an email about it. I sent you the links for the sessions. I think they're all on Zoom. I'd have to double check to make sure that they are. I think they are. But they're not the same Zoom links as we're using for this class. It's a Zoom link that's being used for the whole day of the candidate interviews. And so someone else is managing that. I'll be logging on just the same as you do to observe that, right? I won't be controlling it. But I'll give you another reminder towards the end of next week that that's happening, and you know, a refresh of instructions and stuff. So anyway, that's kind of the plan for today. Kind of finish up the Mendelian genetic stuff and then jump into the math, which is always a potential quagmire of, you know, logic and stuff. All right, so essentially, as I said already in response to Tanya's question, dihybrid cross is in many ways the same as a monohybrid cross. It has the same structure, it has the same kind of formal progression through 3 generations. And as you can see here, it has the same characteristic where the P generation is true breeding on both sides, right? That's super, super important, because otherwise you're not going to get out what you expect to get, right? It would really screw things up. So whenever I'd say to you, okay, we've got a dihybrid cross, blah, 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 you're like, okay, I know exactly what I'm dealing with here. There's no tricks or surprises. I know it's going to be this, 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 and this, right? And so just to give you a heads up for the exam, I will be giving you most likely dihybrid cross-based questions. Maybe giving you monohybrid ones, just kind of like easy ones because they're pretty straightforward. So you do need to know how to do a dihybrid cross, and also I will be giving you chi-square problems based on dihybrid crosses and other crosses, right? So you need to very much, and I'm not, I guess I'm doing you a favor by telling you in some ways what's going to be in the exam, but I'm really not telling you how to do it. right, in the exam, but you telling you that you do need to know how to be able to answer those questions, right? Those are going to be some of those higher level processing questions that you need to be able to do to be, get a really good grade in the exam, right? So give you that heads up. So between now and the exam, you should be practicing dihybrid crosses, maybe even test crosses, back crosses, and certainly you need to be testing yourself on chi-squared problems, right? Because you will need to do that in the exam, right? So giving you a full heads up there ahead of time. Anyway, so we have and just by convention, we always have the dominant, and the recessive, like this, right? We tend not to give you the dominant recessive and recessive dominant just because it messes with your heads. And so these are our traits: seed color, seed shape. Phenotypes are round versus wrinkled for seed shape, yellow versus green for seed color. Right, so now we have potentially we will have 4 different combinations of these phenotypes, right? Because you can have round and yellow, wrinkled and green. Those are called the parental combinations, right? Because you see them in the parents. And then you can have the round and green and the wrinkled and yellow, right? So you can have 4 in total. So, question at the top. What are the genotypes first going to be for the F1s of this cross?
Big
So,
R, small r, big Y, little y.
correct. So someone else, thank you, Tanya. Someone else, oh, Kim, I'm just going to go through this and then I'll answer your question, okay? So someone else, can you tell me what the phenotypes? Yeah, round yellow, same as the dominant, right? Because they're heterozygotes, and so they're going to be the same phenotype as the dominant, homozygous dominant parent was. Awesome, cool. So it's exactly the same as a monohybrid cross, but now we're just dealing with two traits rather than just one. Kim, I'm just going to bump back to Kim's question. We will talk about this as we go through, for sure. And let's just double check the schedule. Okay, so unfortunately, because we have this invited lecture, we don't have a kind of a recap or catch up day before the exam. I was trying to think how. So essentially, what you're going to be getting is a 40 question, and we'll talk about this again a little bit closer to the time too. You're going to be getting a 40 question exam. You're going to have an hour and 15 minutes in which to do it, which isn't a huge amount of time, right? So even though you can use Google and like to look up questions, you don't want to rely on that because it's going to eat away at the time that you have to answer those questions. One key point, which is worth reiterating, I think well, I'll definitely put it in the description of the exam, is, the questions come up one at a time. And the order in which you get the questions is randomized. So everybody is going to have essentially a different, I'll get to you in a second, Rose, is going to have a different order of questions, right? And so you can't leave questions and go back to them later. That's one of the big differences between paper exams and doing these exams online. So you have to answer that question best as you can, and as soon as you go on to the next one, that's it, it's done, right? So another good point that Rose brought up, you can take this at any time during the day. You can up to and include, well, not including, up to 11:59 at night on, what is it, the 16th of February. Obviously, you'd want to start it earlier than 11:59 at night because that wouldn't give you any time to do it. So give yourself at least an hour and a half before the deadline to start it. I will be actually kind of sitting here on my couch during at least the first half hour of that lecture period on Zoom, most likely on my own. If any of you, while you're taking the exam, have any questions, right? So I'm probably not going to be here for the whole hour and a quarter just because there's only so much BBC News that I can read. But I'll be here for at least half an hour, 45 minutes, so that you have that option to talk to me directly if you have a question. So there's that as a potential benefit to doing it in the same time as the class period. If you have any problems outside of that time, then shoot me an email and I'll answer them as quickly as I can. Obviously, it depends on where I'm at and what I'm doing, whether I'll be able to do it within 5 minutes or 50 minutes. But I'll make sure I answer those questions within that day as quickly as I can. So getting back to Kim, what to expect. You should expect a lot of basic recall questions, right? Give you one just off the top of my head. What's the genotype of an F1 and a monohybrid cross? 3 answers might be homozygous dominant, homozygous recessive, heterozygous. So that's a simple recall question, right? That should be something that will take you, or should take you, little to no time to answer, and with a high degree of confidence, right? So there are a lot of questions like that. What is thymine replaced with an mRNA? Uracil. I'd probably put down adenine, cytosine, guanine, uracil. You pick uracil, right, for example. So those are basic recall questions. It's just a basic knowledge base that you should have of this information to answer quickly. And those should be easy questions to answer. The hope is you get all of those right. Right? In part because you can just look those up online, although I would not recommend relying on that because it will cut into your time. And in part just because that's stuff that you just should know, right? It's just basic knowledge. From on top of that, I don't know, I can't remember what the percentage would be. I usually aim for around 50%, give or take, of basic recall questions. On top of that, probably 30% or so would be, let's say, things like one of the classic ones that I often use is, if you have a genome that has 20% adenine in it, what percentage of guanine do you have? Right. And so these are questions where you need to do a little bit of a leap, right? So you can't look these up. You have to actually think through them and work them out based on your knowledge. And so if you have 20% adenine, how much guanine would you have? Actually, that's a good question to the class. How would you work through that?
30%?
Right. Is that just a you're good at mental arithmetic thing, or have you come across that question before?
No, well, it's because then you look for the base pair that's going to have the same percent 20, so that leaves you 60% for the other pair,
Right.
and that has to be in half, so that's 30.
Yeah,
That's
exactly.
how I work it. I don't know.
Yeah, that's exactly how you should work it out, right? And so essentially that's a good example of that kind of question because you can't just remember what the answer is, right? You need to remember, oh, adenine base pairs with thymine, right? And so essentially 40% of the genome is going to be A and T, right? So what's left, right? So there's a series of kind of little steps that you have to make to come to the right answer. So that would be a good example of those kind of questions. And then another one, like the next, 20% or so, which is what was 20% of maybe 6 to 8 questions out of the total. Pardon me. I ate my breakfast too fast this morning. Will be questions like, okay, I'll give you a dihybrid cross design, and I'll give you the F2 data, and I'll expect you to work out the chi-square value and the probability of that chi-square test. And I may ask you to give me the probability, like to pick through like a selection of different p-values. I might ask you to make an inference from that p-value. So not only do you have to do the chi-square test, you also have to then tell me whether you think that something is going on or not. So you need a knowledge of what those p-values mean, right? And so those are most definitely the difficult questions, and those are ones that will take longer, right? Some of that is taken into account in, no, they're all equal points. So you don't get more points for the harder ones, unfortunately. Just it would make life a little bit trickier to do in the exam. They all have the same number of points assigned to them as far as I can remember. But you will need to spend a lot longer on those questions to be able to answer them. It's not just a that's the answer type question, right? So there will be a number of those, and there will be some which I will give you some of the information, but you'll need to provide the rest of the information, and then you'll need to go through that. Yeah, I'll try and well, I'll definitely be going through some examples of chi-squares with you, right? And I can just make up some different stuff on the spot, and we'll actually work through those as if I would for an exam question, for sure, Ali. Most definitely. Right, so that's essentially what your exam will consist of. It will be a range of different stuff. Mostly it's going to be multiple choice or multiple question. Multiple answer, sorry. Not multiple question. Obviously it's multiple question. It will be multiple choice or multiple answer. But if it's multiple answer, I'll always tell you that. Usually there's a pick all that apply that will be at the end of the question, right? So you're not left guessing, ooh, is this multiple choice or is this multiple answer kind of thing. I don't want you to waste time thinking about that. I want you to just give me what the answers are. Yeah, good question. And the thing is, I really truly appreciate how nerve-wracking it is that you haven't had an exam with me yet. You don't know really what to expect, and so the first exam is always a bit of a kind of scary moment because you just don't really know truly what to expect until you've done one, right? And it's very hard for me to give you like a mock exam ahead of time without actually giving you an exam, if you see what I mean, right? Because there's only so many things I can ask about in any given section of information. So yeah, I do appreciate that. One thing that I would say is that essentially prepare for the worst, right? Not that I want to scare you, but the exams will be tough. And so, start revising. Well, I don't know, yesterday would be good, maybe a couple of days ago. But whatever you do, don't leave the studying until the last minute. You should be doing this kind of over and over and over again. That's part of why I want to spend the beginning of each class, going over anything that you don't understand from the previous classes, right? Get that repetition, reiteration. That's what the homeworks are for, to kind of, force you to go through that material. That's why, we're doing stuff the way we're doing it. That's why the textbooks are really useful, stuff like that, right? So this is a course that you have to study constantly for, without any shadow of a doubt. Otherwise, you will not succeed, right? This isn't something that you can just, at the last minute, the day before, spend 8 or 9 hours ramming stuff into your head. because that will work really well for the basic recall questions, maybe even a little bit for the, you know, the kind of this slightly harder processing questions. But for everything else, you'll be lost at sea, right? Because those skills take time to develop and to polish, right? So that's a very honest kind of upfront appraisal. It will be a hard exam. You need to study constantly for it to be successful in it. Yeah, and ask me as many questions as you like about the material, right? And those questions should kind of bubble up, right, on their own just by going through that stuff. There will be things that you don't understand on your own, and that's normal, right? That's what I'm here to help you with. Cool. All right, let's get on to slightly more cheerful stuff. It's always a little bit doom and gloom talking about exams, like nothing that anybody ever enjoys, right? Well, maybe there are some people out there that enjoy taking exams, but I certainly didn't when I had to do it. It was one of those kind of, you know, kind of grit one's teeth and get through it kind of deal. Okay, so, but preparation is the key, right? The more prepared you are, the better you will do. And that's something that applies to anything. Job interviews, for example, you know, preparation is key. The last thing you want to do is go into a job interview and not know something about the place that you're interviewing at, right? Because that will look really bad, for example, right? So it's all about just studying the shit out of something. Or as, what's his name? Mark Whitney in The Martian. We're going to science the shit out of this. There you go. It's one of my favorite quotes from that film. Okay, so anyway, tangent aside, good question, Kim. And any, uh, yeah, I like that movie too. There's actually, sorry, kind of go off on a little bit of tangent here. I'll steer us back to the course in a second. The only, because I'm really interested in astrobiology, and actually I was this very morning reading about the Perseverance rover, the Mars 2020 mission that is kind of on its way to Mars as we speak. And the only thing that they got wrong or they exaggerated in The Martian was the force of the wind. So even though Mars has crazy sandstorms, right, which can cover the entire planet, the atmospheric pressure is fairly low. It's about a tenth of the that on Earth, and so there wouldn't have actually been enough atmospheric pressure and wind to push that rocket over. That's the only thing that they exaggerate. Everything else was actually bang on the money science-wise, which is pretty rare for a science fiction film. Anyway, there you go. That's kind of like my little soapbox. I get ever so mad when I watch sci-fi films that break the laws of physics. It just pisses me off no end. Okay, so anyway, getting back to the program. Heterozygous for both F1 generation. Always, 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 right? If so here's another thing to remember. If I'm going to be getting you to do a cross that is not a monohybrid or a dihybrid cross, I will not call it a monohybrid or a dihybrid cross. I'll call it something else, right? And so if I've given you a cross and I don't say this is a dihybrid cross, it isn't, right? It's something else, right? So remember, these assumptions apply only to a monohybrid or dihybrid cross. This particular format is only monohybrid or dihybrid cross. If you were to do a back cross, like we'll be doing in the genetics lab with our little yellow and white-eyed flies, that is very different. That is not a dihybrid cross, even though we're dealing with two genes and each has two different phenotypes, right? It's not a dihybrid cross. So anyway, we get this, and the key here is we have essentially four different alleles, right? We have two different alleles for the seed shape gene, we have 2 different alleles for the seed color gene. How many combinations of alleles are we going to get in our haploid gametes? How many do you think we're going to get?
16.
These are the haploid gametes. Good guess too, Kim, but not right either, which is fine. You're going to have 4, and the reason being is, let's kind of drag out my micro whiteboard. It was funny yesterday because I had to go into work before class for my Wednesday lab. For my Wednesday class for Wednesday lab. I actually gave my lecture in my office, which was kind of weird. But I have this huge whiteboard in my office, which was awesome. I was like a kid in a candy shop. All right, so we have our F1, which is going to be, let's see, let's move this back a little bit. Can you see that okay? Actually, tell you what, I'm going to close my blinds so it's a little bit. Be able to see more. Just have the glare from my front window on the whiteboard. Yeah, blue still sucks though. I'm going to skip using blue, I think. Pardon me. Yeah, that's a little bit easier to see. Yeah, that's a bit better. Okay, so, crap sticks. I've got to close the other one. I'm just going to come home and be like, why are you teaching in the dark? Isn't that kind of weird? Anyway, all right, so a little bit easier to read now, hopefully. So we've got our 2 different genes. Each has 2 different alleles. So when we go through meiosis, and we'll actually get to meiosis in a week or so's time, you'll actually see what happens at a chromosome level in this, which is why this is going to essentially why this gene, the 2 alleles are going to segregate randomly, and why that segregation of the alleles for this gene is going to be completely independent of the alleles for this gene, right? That's the law of independent assortment that we talked about last class. And so you can have each gene right? So for the R gene, for example, you can have either a gamete with either an R or a Y, right? Makes sense. For your Y, you also get either a big Y or a little y, right? But that doesn't just occur twice, because you can also have your big R, oops, and little r over here, and your little y and your big Y. Because you have 2 different genes. So each allele is going to have, each gamete is going to have an allele from each gene, right? So wherever you have a big R, you're going to have either a big Y, which is this one here, or a little y, right? Every time you have a little r, you're going to have a little y or a big Y. Or you could do it the other way around. You know, every time you have a big Y, you're going to have either a big R or little r, for example. It doesn't really matter which way around you do it. It's exactly the same outcome. And so essentially, you're going to have 2 to the power of 2 combinations. If you do a tri-hybrid cross, what would that be? That would be 2 to the power of 3. That'd be 2 times 2 times 2. That'd be 8 different combinations. If you did a quad hybrid, well, that's just getting crazy now. That'd be 16. I know it kind of gets a little bit nuts when you start getting above 2. I think I did a tri-hybrid cross once. It was pretty crazy. That's why I didn't do it again. All right, does that make sense? That's why you get the that's why you get the 4 different gametes and 4 different allele combinations. Quick question from Kim. This example similar to the chi-squared test. Well, the chi-squared test is really, we will be using this for the chi-squared test, right, to illustrate it, because it's a very clear example where we have defined expectations. But a chi-squared test can be for anything where you expect a certain outcome and you wish to test whether or not your actual outcome fits that, right? So it could be simple thing as, you know, if you have a litter of puppies and you got 8 puppies, you'd expect there'd be 4 boys and 4 girls, right? You could use a chi-squared test to see whether or not that expectation has been met, whether your essentially whether your prediction is a good fit. Let me just take a picture of this before we move on. Hopefully my camera behaves itself. All right, that'll do. Okay, now probably Tanya, where you're getting the 16 from is the F2 generation, right? So each individual of the F1 generation, right? Because we have, where are my fingers? We've got 2 parents, obviously. Each one's going to be heterozygous. So each parent contributes 4 potential gametes, right? And because segregation is random, and it's random for each of those 2 genes, we're going to have those 4 potential gametes in equal proportion, right? 1 to 1 to 1 to 1 ratio, right? Because essentially we have a 25% chance of finding either one of those in, like if we were to pick one of them out of a pool, let's say, we'd have 25% chance of finding any given allele combination, right? And so that's where this is going to be a little bit crazy, but we'll try and make it fit. If you write those out, let's make sure I do this correctly, right? These will be the gametes provided by parent number 1, let's say, right? Parent number 2 is exactly the same because it's also a heterozygote from the same generation. So it's also going to be providing exactly the same gametes or potential gametes, right? We don't know which ones are going to actually take part in fertilization, but we assume that they're going to have equal chance of doing so, right? Then we're going to have the same here. Let's see. Yeah, you can actually see this. This isn't quite as terrible as I thought it would be doing it this way, right? That's your 16 possible combinations, like so. right? And so one thing I try and stress, whatever level I teach this at, is that this isn't rocket science, thankfully, you know, because I wouldn't be very good at it if it was. It's quite simple. You just simply have to follow each step through the process, and you won't make any mistakes, right? So don't jump to any conclusions. Don't kind of, unless you know this off the top of your head, right? Simply work through this and you will not get it wrong. The same applies, right? So I'm not going to fill this out because you can do that. It's really straightforward. You just add these together. So you just get, right, that's homozygous dominant, that's a parental, it's going to have be dominant phenotype wise, right? And for example, where are we at? We'd have to go down here somewhere, apart from that bloody fly. Let's look. So this one, for example, hey, look at that. In like the Karate Kid had a little annoying fruit fly flying around. Right, so this one, for example, is going to be a combination of the parental phenotypes because it's going to have round seeds and it's going to be whatever it's going to be green, round green seeds, right, which is a combination that you do not see in the parents, right? And you can just go and fill this whole thing out. Now, what you can also do is, you know, just to actually, before I move on to anything else, does anybody have any questions about any of this? Trying to make sure you can see as much of it as possible. Hopefully it should make sense. It is relatively straightforward. You just have to follow it through, right? So one thing I'd actually recommend you doing is work through this. You can actually make up your own chi-square, your own dihybrid cross if you wish, right? I always find it fun using seven dwarfs from seven white, right? So you could have one that's grumpy and one that's, I don't know, whatever the other different, one that's grumpy and one that's happy, you know, and whatever other traits you want to throw in there and actually work through it. You'll get exactly the same answer as you would with this particular one here, right? So one thing that you're going to do though is when you go through all of this, you'll always find this one down here. That's our one double homozygous recessive, right? And then scattered around here, you'll find the two threes, and then the rest of it will be the nines. So you'll always, when I'm just going to rub this out. Actually, let's take a picture of it first. Where's the camera go? You'll always get, once you add all of these up, I'm just going to rub this out on the bottom so I have space. If you write out the different combinations of phenotypes, so it's going to be, what is it, round, yellow, round, green, wrinkled, if I can spell that right, that's about right, yellow, and wrinkled green, right? Those are the 4 different categories of phenotypes. Again, these ones are new combinations that you do not see in the parents, right? That's actually one of the interesting things about dihybrid cross. In the monohybrid cross, you only see whatever phenotypes you see in the parents. Dihybrid cross, you'll now see two combinations which are not present in the parents, right? And that ratio will always be 9 to 3 to 3 to 1, right? Always, always, always. If it's not a 9 to 3 to 3 to 1 ratio, then something else is going on. Your assumption about how the cross was organized and laid out is wrong. And actually coming back to Kim, these numbers, these ratios, these are your predictions, right, from which you'll calculate your expected values for a chi-squared test. Right, so if you were to, I always use easy numbers, right, because it's easy math to do in my head, right? This all adds up to 16. So essentially 9/16 of the seeds produced, or whatever peas produced, will be round and yellow, right? And so if you scored 160 peas, you would expect that 90 of them would be round and yellow, 30 would be round and green, 30 wrinkled and yellow, and 10 of them will be wrinkled and green. Those would be the expected values that we would use for your chi-squared test. Obviously, the actual numbers may be different. Maybe 93, 27, 28, and 12, for example, right? So that's where you would use your chi-squared test to see if those small differences are explained just by random chance, or if there's something funky going on which you're not taking account of. Does anybody have any questions about any of that stuff? No. So what would happen? Here's a good question, make you think. What would happen if you crossed a heterozygote for both of these genes with a pea plant, whatever, that is homozygous dominant for both? What ratio would you get in the offspring? So this isn't a dihybrid cross, obviously, because you wouldn't have a homozygous dominant in the F1. But if you did a cross that was between a homozygous dominant for both genes with an individual that's heterozygous for both genes, what ratio of phenotype combinations would you expect in the offspring? Think about how many alleles each parent is going to contribute to the next generation. This is also often an exam question that comes up. All right, shall I give you a little bit of a hint? They would not all be dominant. That's a good guess though, Motaz. Because if you have actually, shit bits, I said the wrong thing. You are correct. Yeah, you're totally right, Motaz. Awesome. Sorry, let me change that question. I'm sorry. That's my mistake. Good job. You caught me out on that one. All right, that's what I should have said. Okay, so let's just kind of wind back that Zoom session and pretend I didn't make that mistake. What if you cross a individual that is, oh, here you go. This is like a test cross, right? Essentially. Your tester is homozygous recessive for both. Actually, I've got to put a little message in there for Motaz. Where is that? There you go. Just put that on the record. If you had a homozygous recessive tester, right, which is homozygous recessive for both, what phenotype ratio would you expect in the offspring if the other parent was heterozygous for both genes? Holding up a big clue for you. Just here. There's going to be 4 different phenotype combinations. That is correct. What will the ratio be? Yeah, totally. Thank you, Motaz. Got it right twice in a row. It's going to be a 1-to-1-to-1-to-1 ratio, right? Because you're only going to get bloody flies. Don't know where they come from. You only get 1 possible gamete from your homozygous recessive individual. And from your heterozygote, you get 4 possible gametes, right? Which have an equal probability of being produced. So you'll always get, or you should get, a 1-to-1-to-1 ratio of the phenotype combinations, because these are all going to be different. You're going to have dom-dom. I'm just going to use D. This is going to be dom recessive, recessive dominant, and recessive recessive, right? So that's just another way of thinking through this kind of problem. Actually, this would be what a test cross for a individual that you think is heterozygous for 2 different genes would look like. Let's see if I can get. If I had any virtual chocolates to throw your way, Motaz and Rose, I would do so, but I don't. So I'm gonna. If I did have any, I'd actually probably just eat them myself. I really like chocolate. Okay. Anybody have any questions about any of that jazz? Right. And one thing too, as well. So this is again, I try and because I know what's going to be coming up for the rest of the course. I can give you a heads up essentially about other things that are going to build on this. And so one of the things that builds on this, right, either this kind of stuff or the dihybrid cross, anytime you get fewer of these non-parental combinations, that shows you that these genes are not assorting independently, right? So our assumption is that they are, right? Hence the law of independent assortment. But if those genes are actually fairly close together, they will not behave, or if they're on the same chromosome and they're reasonably close together, they will not assort independently. And so you'll see an excess of the parents and a lower number of they're called recombinants in this case, but the non-parental combinations, right? So if you see a deviation from this ratio, if it's a test cross, or the 9-to-3 to 3-to-1 ratio, if it's a dihybrid cross, then that deviation is caused by a few different things, right? Either the phenotypes are affecting each other, which is called epistasis, or the genes are not assorting independently, right? And so that's one of the ways in which these kinds of tests start becoming useful, right? That's not something we're going to cover right now, and actually we're not going to cover that until the second section of the course from memory, right? So it's not something that's going to come up in exam 1, but it's worth, you know, sticking that in your head and then pulling it out later. Cool. Anybody have any questions about any monohybrid or dihybrid cross? Because otherwise we're going to skip on to the math. Yay. That's so funny. It's really funny when I say completely the wrong thing. I'm like, no, you're wrong. Oh shit, actually you're right. That always makes me laugh when that happens. All right. Speak now or hold your peace for at least, you know, a little while. So, where are we at? Yeah. Well, I mean, everybody makes mistakes, right? And actually, that was sometimes I often find when I make mistakes that actually turns into something cool to talk about in terms of what we're learning. So yeah, it's like, oh right, yeah, you know, you kind of like work through it. It's like, oh, I was wrong. That's actually one of the things I like a lot about uh, genetics, right? Because it is so kind of logic based in many ways that you can quite often figure out, oh, actually I was wrong, right? Because you know you kind of go through stuff using the same assumptions that you should do normally, and then something doesn't fit, and then it's like, oh, okay, you know, I was wrong. I actually had a very brief tangent. I was postdoc at Penn State um, many moons ago, and I was doing some work on cell death and survival in this particular mutant. And I thought I found out some really cool stuff, right? So I found these cool uh, data, and I prepared an abstract for this conference. Like it's called a worm conference, right? It's where everybody that works on worms goes to talk about their stuff. It's really huge, like 2,000 people or so at UCLA every 2 years. And so I was super excited about going to that with my first one that I'm going to, and all that kind of stuff. And then I realized that actually I scored the cells wrong, and so I wasn't seeing an effect. So I had to withdraw the conference abstract, and I didn't go to the conference. I ended up going to a small regional one in Philadelphia instead, which was fun, but it wasn't quite the same thing. But that saved me a whole ton of potential grief chasing down the wrong rabbit hole. So it actually worked out pretty well. But at the time, I was like, "Oh, this sucks." But you know, it's yeah, or even worse afterwards, right? Because once you have that stuff out in the scientific domain, it becomes, you know, part of collective knowledge. And if you find out that you're actually wrong, it's far more embarrassing to go back and actually say, well, sorry, everybody, kind of screwed up on that one. That was bullshit. Uh, Here, read this paper that I just wrote. You know, it doesn't really sound good. So anyway, that's why you have to be very, very careful, like double checking your work, basically. Anyway, so what we're going to do now is we're going to at least make a little dent in this. And so because Mendelian genetics or transmission genetics, whatever you want to call it, is very much a matter of probability, right? It's not a matter of certainty, right? It's a matter of probability. Pardon me. It's really important to understand how we work with those probabilities. And particularly because in many instances, we're really interested in multiple, like the interaction of multiple probabilities. You know, if, for example, you're a couple in your 40s, right, and you want to have children, what's the probability that you'll have a child that has Down syndrome, right? What's the probability if you have a second child that both children will have Down syndrome, right? If you're an Ashkenazi Jew, for example, and which has a very high prevalence of Tay-Sachs disease, right? what's the probability that if you're both heterozygous for this, your offspring will have Tay Sachs, which is a really horrible disease. Children tend to die when they're 4 or 5 if they have it, right? And that's actually another aside. Church of the Latter-day Saints and I don't know what you'd call it, the Jewish community have the 2 best genealogical databases in the world, which is kind of neat. And one of the reasons for I mean, there's other reasons. Part of it is if you want to become an Israeli citizen, you have to prove that you're Jewish on your mother's side. But also because there's a bunch of diseases that are very highly prevalent in Jewish communities, right? And so if you want to have children, then it's good to know whether or not you're a carrier for a particular disease, right? So there's a lot of instances where these probabilities actually become really important, right? And again, they're not certainties, they're probabilities, right? So they're really just possible estimates. And so there's a couple of different things to think about when it comes to probability, right? One is, are these events independent, or are they exclusive? Sorry, just listen to the front door cycle, right? Because they do you deal with them in very different ways depending on that, right? So an independent event is really something that if you have one event, you have an X probability, right? Whatever result you get for that one event has nothing to do if that event is repeated. And so each of those events are independent from each other, right? The classic example is flipping a coin, right? Whichever head or tail you get for flip number 1 has absolutely nothing to do with what you get for flip number 2 or 3 or 4 or whatever. And it's very much the same for having children, right? Whatever sex your first child is has absolutely no effect, at least biologically, on what the sex of your second child is going to be. I mean, I know like some families have like real high prevalence of boys or girls. I mean, both my brother and we have 4 girls between us, which is kind of weird. Um, I don't know what that's about, but it's just, in most cases, as far as I'm aware, probability, random chance, right? And so those events are independent. Sorry, just seeing what you wrote there. Okay. Oh, wow. So there must be something going on, right, that actually affects her, because I've heard of examples where you have like, stop doing that. I'm going to turn this round and show my class what you're doing. My wife's trying to distract me. Anyway, getting back to the program. Yeah, I've actually heard of cases like that, and so it's neat to hear one about that from you as well. So there must be something going on there, but what that something is, I don't actually know what it is, right? But there's something that modifies those probabilities, right? An interesting question, actually. And then you have, oh, cool, boy and then a girl, and then another boy. Wow, that'll keep you busy. Yeah, we've just got the two girls, and they're both teenagers or nearly teenagers, so that's keeping us busy in other ways. And then you have events which are mutually exclusive, right? So if you have a boy for your firstborn, like Tanya, then you can't have had a girl for your firstborn, right? Because they're exclusive, right? It's either a boy or it's a girl, right? You can't have both at the same kind of time, unless you have twins, but that just gets kind of all kinds of irritating, right? And so how we deal with those events differs. So when events are independent and you have multiple events occurring, if you want to find out the probability of that string of events, right? So if you want to find out the probability of having two girls, for example, right? Then actually, cool, I can use Tanya's children as a learning moment as well. Get to that in a second. If you want to have, or if you want to know the probability of having two girls, then you take the probability of each of those, which is a half, and then you multiply them, right? So the probability of having two girls is half times a half, which is a quarter. Does that make sense? So only if you had a whole bunch of couples together, right? If say you had enough of them and they all had two children, then you'd expect to find only a quarter of those couples have two girls, right? Similarly, you'd expect to have only a quarter of the couples have two boys. Now, why would you have half of the couples having a boy and a girl? When you think about that. That actually comes to what we'll be studying in a little bit on another slide. So why would half of the couples, let's say, that have two children, why would half of them have a boy and a girl? Right, if the probability of having a boy and a boy is a quarter and a girl is a quarter.
Would it be because you also multiply? It's going to be 25, but it's like boy, girl, and it can be like the other sequence, girl, boy.
Correct.
And you add those?
You add them because they are mutually exclusive. Right, so if you have a couple that had a boy and then a girl, you have a probability of a quarter, right? Because it's half times a half. But then you could also have a couple that has a girl and then a boy. That probability is also a quarter. But you can't have a couple that has both. So you add a quarter to a quarter, that gets you a half, right? And one of the kind of neat checks is, okay, all of these, right? If we have two possible outcomes for two events, all the probabilities should add up to 1. And so that's like a neat kind of reality check, I guess you could call it, or work check. Do my probabilities add up to 1? And they should do. If they don't, then there's something going on that you haven't taken account of. Right? So what would be the probability that, like Tanya had 3 children, what would be the probability that you'd have 2 boys and a girl? Sorry, I told you I was going to use you as an example.
1/8.
Exactly. Why? How did you get to that? Explain it to
'Cause
everybody
it's
else.
just- okay, so the possibility of having a boy or a girl is 1/2, and so it'd just be 1/2 for the first time, 1/2 for the second time, and then 1/2. It's just 1/2, 1/2, 1/2 multiplied by each other, and that's 1/8.
Exactly, right? So every time you have another, you just again times it by the probability of each individual event, right? So you can see that if you had 5 kids, right, the probability of having 5 girls is it's actually really, really quite small, right? Because it'd be a half times a half times a half times a half times a half, which would be 1/32, whatever that is in decimal. Right. So only if, you know, out of all of the families in the US that have 5 kids, only 1 in 32 will have 5 girls. And essentially what you're saying is that, okay, every time I do this, the odds of getting a certain combination gets smaller and smaller and smaller, right? So if you have like a winning hand at poker, right, by random chance, then the probability that you'll get another winning hand, right, it gets smaller the more times you do this, right? If it is random, the events that are controlling that particular outcome. Does that make sense? Anybody have any questions? Cool. So then we can, we've kind of already hit, yeah, go on it, hit this, but this is another thing that's important to consider, right? Because we're going to need to use this when it comes to thinking about order, right? The order that things can occur in, right? So if we have like a firstborn child has a particular disease, right? You know, what's the probability that the second child will also have that, right? If we have two children, things like that, right? So mutually exclusive events are important in figuring out also that kind of order deal, right? And so that basically means that if you have one event occur, you cannot have another event occur, right? So if these occur multiple times, or oh, I think a dog got caught in the door, or sorry, temporarily distracted. Um, gotta get my train of thought back. It's just one of the puppies got caught in the back door when my wife was going out. It's a little hard to kind of get my train of thought back. So yeah, so or if you have a range of possible outcomes, right, then the probability of getting more than one of those outcomes, because they're exclusive, you have to add those together, right? So the example here is neat, like a 20-sided die. You know, if you play Dungeons and Dragons, that might be familiar. But if you have a 20-sided die and you need to get two particular numbers, like what are the odds of getting that number? Well, you can only get one number or another. You can't get both of them, right? And so essentially, if you're going to be getting one number or another, then you're going to be getting two out of the total possible, right? So your probability is actually 2/20ths or 1/10. It could either be this number or this number. You can't have both, but because you could have either one, the probability of getting one of them is the probability of getting either of them added together. Right, and so when you have mutually exclusive events, the probability of getting any one of those actually increases. And we're kind of just about run out of time. So what we're going to do at the beginning of the next class, we're actually going to go through, it's actually kind of neat using the dihybrid cross to talk about that. And we'll actually go through this and you'll see why this applies to, for example, a dihybrid cross. And also we'll quickly go through order of events, which is also a kind of fun one to think about, right? Once we do that, and that shouldn't take super long, right? Give or take, then we'll go through chi-squared tests. So before class on, what's the day today? Yeah, Tuesday. Read through this stuff, right? Have a quick look through the material on goodness of fit, the chi-squared test, right? So you have some idea like what exactly are we working with here. So when we hit it in class, I can spend a little bit less time on that and a little bit more time on doing examples. So you get to practice it, right? Which is what's important. Cool. All right, we've hit the limit. So I'm going to say goodbye. Wish you all a fabulous weekend. And I shall see you on Tuesday. If you have any questions about any stuff that we've covered today, and thank you everybody for your own stories. It's always super neat to hear about that. Then please let me know by email and I'll get back to you as soon as I can. If not, bring those to class on Tuesday. All right, I'm going to go see what happened to that puppy. So catch you all later.